Hey there, science fans! Today, we're diving into the fascinating world of photoelectric colorimeters. These nifty instruments are like the detectives of the chemistry lab, helping us understand the secrets hidden within solutions. But how do they work? It's all about light. Imagine shining a light through a colored liquid. Some of that light will pass through, while some gets absorbed. A photoelectric colorimeter measures how much light makes it to the other side. By analyzing this, we can learn about the concentration of substances in the solution. These instruments are used in various fields, from chemistry and biology to environmental science and medicine. They're essential tools for analyzing water quality, studying chemical reactions, and even diagnosing diseases. So, are you ready to learn more about these amazing analytical tools? Let's get started. The first key component of a photoelectric colorimeter is its light source. Think of it like a flashlight beaming light through our solution. Commonly, we use a tungsten filament bulb for this purpose. It's similar to what you might find in an old-fashioned incandescent light bulb. For more specialized work, we might employ a quartz lamp, which produces a more intense and focused beam. Now, not all light is created equal. Different substances absorb light best at specific wavelengths. That's where filters come in. These clever components select a specific wavelength of light to shine through our sample. We have two main types of filters: color filters and interference filters. Color filters work by absorbing certain wavelengths of light and allowing others to pass through, much like stained glass. Interference filters are a bit more complex. They use the principles of light interference to select a very narrow band of wavelengths, ensuring we're only measuring the light that matters most for our analysis. Now, we need something to hold our precious samples. Enter the cuvette. These small, rectangular containers are designed specifically for use in colorimeters. Cuvettes are typically made of glass or quartz, depending on the type of analysis being performed. Glass cuvettes are great for measurements in the visible light range, while quartz cuvettes are used for measurements in the ultraviolet, UV, and visible regions. Why is the material important? Well, we don't want the cuvette itself to interfere with our measurements. The material needs to be transparent to the wavelengths of light we're using for our analysis. Section 4. From light to electricity, the photocell. Remember how some light passes through our sample? We need a way to measure that. That's where the photocell comes in. This ingenious component converts light energy into electrical energy. Think of the photocell as a solar panel, but on a much smaller scale. When light strikes the surface of the photocell, it knocks loose electrons, creating a tiny electric current. The more light that hits the photocell, the stronger the current. This electrical signal is what allows us to quantify the amount of light that passed through our sample, giving us valuable information about its composition. Section 5. Measuring light intensity, the galvanometer. We've got our electrical signal from the photocell, but how do we actually read it? That's where the galvanometer comes in, the final key component of our photoelectric colorimeter. The galvanometer acts like a sensitive meter, measuring the strength of the electrical current generated by the photocell. This reading is directly proportional to the intensity of light hitting the photocell. In simpler terms, the brighter the light, the higher the reading on the galvanometer. This reading allows us to calculate the transmittance and absorbance of our sample, which are key parameters in colorimetric analysis. Section 6. Using the colorimeter, a step-by-step guide. Now that we understand the main parts of a photoelectric colorimeter, let's see how we actually use one. First, we calibrate the instrument using a blank solution. This solution contains everything except the substance we want to measure. This step sets our baseline. Next, we place our sample in a clean cuvette and insert it into the colorimeter. The instrument shines light through the sample and measures how much light passes through. The colorimeter displays the reading, usually as transmittance or absorbance. Transmittance tells us how much light passed through the sample, while absorbance tells us how much light was absorbed. We can then use the Beer-Lambert law to determine the concentration of our substance of interest. This law states that absorbance is directly proportional to the concentration of the analyte and the path length of the light beam through the sample. Finally, we interpret our results, comparing them to known standards to determine the concentration of our substance. And there you have it. With the help of a photoelectric colorimeter, we can unlock the secrets hidden within solutions and gain a deeper understanding of the world around us.